Welcome to the Chemistry, Biology and Math Revision Hub. Today we are doing the Pearson Edexcel International A-Level Biology Unit 6 for October 2024. Let's begin with the first question. In question 1, Gibberellin stimulates germinating wheat grains to synthesize amylase. State why germinating wheat grains need to produce amylase. This is because amylase is required to break down starch from the endosperm into maltose and later into glucose that can be used in respiration. So I said amylase breaks down starch stored in the endosperm into maltose, which provide energy for growth and development of the embryo. Then in part B, the drawing shows a wheat grain and locations of the endosperm in the embryo. So we see the embryo here and then the endosperm the other side. Describe an experiment to determine if gibberellin stimulates the synthesis of amylase by the endosperm or the embryo of a wheat grain. So here we will cut and obtain the embryo alone and the other side the endosperm, and then we'll expose both to gibberellin and then place them onto a petri dish containing starch and see if digestion of starch takes place. And of course we'll include control experiments. So I said use aseptic techniques. Prepare several groups of wheat grains and carefully dissect them to separate the embryos from the endosperm. And then set up 4 test conditions, like could be Group A, the endosperm only plus gibberellin solution, Group B, endosperm only plus water, which is the control, and then Group C, the embryo only plus gibberellin, and D, embryo only plus water, again, this being another type of control. Then add starch agar to petri dishes and place each sample on the agar. Incubate all groups at the same temperature for 24 hours, and then add iodine and observe the clear zones around the sample. The presence of a clear zone indicates starch breakdown by amylase. Then we compare the sizes of the clear zones to determine which tissue, either the endosperm or the embryo, is producing amylase in response to gibberellin. Then in part C, describe how gibberellin increases amylase synthesis. It could be acting as transcription factors, so I say gibberellin binds to receptors in the oluron layer of the seed. This activates transcription of the gene encoding amylase. Messenger RNA is translated into amylase enzyme, which is then secreted to hydrolyze starch in the endosperm. In question 2, the photograph shows Kenaf plants that grow in many African countries. So we see them here. They say the plants are grown for food and as a source of fiber. A scientist investigated the tensile strength of samples of fiber from Kenaf plants. Some of the samples of fiber were treated with hydrogen peroxide and other samples were untreated. So here we see the force needed to break each fiber was measured. The diagram shows the apparatus used. This is the force meter, then the fiber is placed there. There is a computer for precision measurement of the results. Then they say the apparatus shows a force meter that pulls on the attached fiber. The force needed to break the fiber is recorded on the computer. They say describe how the scientists could reduce one risk when using the apparatus. The risk could be the operator could be hurt, like the fibers could snap and then they hit them, so the fiber may snap suddenly and cause injury to the eyes or the skin. How the scientists could reduce this? They wear safety goggles and ensure the fiber is securely clamped and the apparatus is operated behind a safety screen or at a distance. Then in part B, the table shows the results of this investigation. We see the forces needed when treated with hydrogen peroxide and the other side when untreated with hydrogen peroxide. And they say calculate the mean percentage increase in tensile strength of fibers treated with hydrogen peroxide compared to the untreated fibers. They want the answer to be in 3 significant figures. Together mean just add up everything here and divide by 6 because there were 6 samples. So that is going to be the mean. So the percentage increase is that minus that, divide by that, times 100, giving us 10.4%. Next, they say some variables were controlled in this investigation. State one abiotic and one biotic variable other than temperature that could affect this investigation. The abiotic, pH, it could be concentration of hydrogen peroxide and more. And the biotic, these are to do with the fibers, so age of the plants or the fibers. Diameter of the fibers, you could even use length of the fibers. Then choose one of the variables you have identified in part 1. State how this variable can be controlled. The age of the Kenaf plant. How can we control use fibers collected from the same plant? are all plants of the same age that are grown under similar conditions. Then part D says sclerenchyma fibers strengthen plant stems. Describe the structure of a sclerenchyma fiber. Sclerenchyma fibers are dead, hollow, and lignified. So I said sclerenchyma fibers have thick lignified cell walls that make them strong and rigid. The cells are dead at maturity with a narrow lumen and hollow interior. They possess end walls and often contain pits. In question 3, the photograph shows pea seeds with growing roots. We can see them there. They say mitosis occurs in growing roots. Scientists allowed pea seeds to start growing in different concentrations of oxygen. Pea seeds were grown in oxygen concentration of 5% and 20%. After 2 days, the root tips were removed. The tips were stained and observed under a microscope. The mitotic index for each root tip was determined. So the results are given here, the different concentrations or percentages of oxygen. The first question says, state a suitable null hypothesis for this investigation. Since we are comparing experiments at 2 different percentages of oxygen, so we can say there is no significant difference between the mitotic index of the pea root tips at 5% and 20% of oxygen. The next they say draw a suitable table to display the data and your calculated means. So here I just use the values. I place them initially as they were in the corresponding tables as raw data. And then I got the means. For the 20% oxygen, the mean is 5.7. And here the mean is 4.0. So basically add up all these and divide by 9. Then add up that and divide by 9. You get that. That's how the table is supposed to be. Key thing, put labels and units. Then in part C, they say draw a suitable graph to show the mean mitotic index after 2 days in 20% oxygen and the mean mitotic index after 2 days in 5% oxygen. They want us to include an indication of variability. That means we need to place error bars. So I need to find the maximum at each and the minimum. Here we have the mean as 5.7 and the maximum is about 6.2 here. Then the minimum is about 5.1, so 6.2, 5.1. And here the mean is 4.0, the minimum is about 3.6, and the maximum is about 4.4. So to draw my graph, I drew a bar graph, and as you can see here we have the concentration of oxygen in percentage and the mean mitotic index, since the values were showing at the mean values and then just the error bars, so I used mean mitotic index. So I included the lowest here is 5.1, maximum 6.2, and here the minimum was 3.6, maximum was 4.4. The error bars are placed appropriately. Actually, there is also no overlap, so the results are significantly different. Then in part D, the scientists analyze the data with a t-test using the formula, this one here, where this is the mean value of each treatment and is the number of the samples. We've seen that is 9. And then they've given us the SA and SB values. They want us to calculate the value of T. I'm going to assume this value is for the 20%. Nobody cares. You can switch them around. If you get the negative answer, or for T, you can ignore it. So you can use A for whichever and B for whichever. It doesn't matter. So my T is going to be the mean. If this is my A, then the mean is 5.7 minus the mean at this point, which is 4.0. Divide by the square root of this value here, divide by 9 plus that value, divide by 9, because 9 is the number of samples. And using a calculator, I got 11.33 as my calculated value of T. Then next they say the table shows the critical values of T for different degrees of freedom. The number of degree of freedom should be NA, which is 9 minus 1 plus 9 minus 1, giving us 16. So if 16, it means we're going to go at 16 at P equals 0.05 and find the critical value, in this case 2.12. So they say describe the conclusion that can be drawn from this investigation. Use the information in your graph and the table to support your answer. From the graph, we see there is no overlap, meaning they are significantly different. And from this table, we see the calculated value and the critical value. The calculated value is way higher than the critical value, so we reject the null hypothesis. So I say the calculated T value, which is 11.33, is greater than the critical value 2.12. Therefore, the null hypothesis is rejected, meaning The difference in mitotic index between the two oxygen concentrations is statistically significant. The pea seeds grown in 20% oxygen concentration have a higher mitotic index than those grown in 5% oxygen. And also there is no overlap in the error bars, so the results are significantly different. Then describe how the scientists could extend this investigation to collect more data to either support or reject the null hypothesis. Repeat the experiment maybe at more oxygen concentrations. You could repeat the experiment using more sample size or using different types of plants and so on. So I say the scientists could repeat the investigation with a larger sample size to improve reliability. They could also test additional oxygen concentrations to observe a trend and threshold effect. They could also use different temperatures. In question 4, the photograph shows the pondweed Elodea canadensis, which is this one here. They say this plant grows in ponds and rivers throughout North America. A student read that pondweed grows fastest near the water surface. The student formed the following hypothesis. The farther away from the water surface, the lower the rate of photosynthesis as less light can be absorbed by the pondweed. So basically it's like a light intensity. Plan a laboratory investigation that can be carried out to find evidence to support or reject this hypothesis. Describe the preliminary practical work that you might undertake to ensure your proposed method would provide quantitative results. In the preliminary practical work, we usually find out how we're going to vary the independent variable, how we're going to measure the dependent variable, how we're going to control variables that could change, and so on. So I said find suitable growth conditions like temperature, light intensity, carbon dioxide concentration, find suitable pondweed to be used, find a suitable method to measure the results. In this case, it could be volume of oxygen produced, how are you going to set up the photosynthesis and so on. Then find a suitable method to vary the depth of water, and then find ways to control other variables like light intensity, pH, temperature during the experiment. In part B, devise a detailed method including how you would control and monitor important variables. So here I say that dependent variable is the volume of oxygen released per unit time because this is telling us about the rate. Then you set up a photosynthesis with a healthy Elodea plant in it. Actually, if I show you, I drew something here. Photosynthesis could be like this. But of course, I used a smaller test tube. But if you're working with a bigger tank or you're varying the depth, you could clamp the pondweed so that you can vary the depth. Or you could use this kind of setup. Then the volume of the gas, which is oxygen produced, is going to be measured or collected using a syringe. And then you can calculate the rate by using the volume produced per specific time in order to get your answers. So use a clamp to hold the Elodea at different measurable depths. Could be 0 centimeters, 5 centimeters, 10 centimeters, 15 and 20. You shine a lamp above the experimental setup. Keep the distance between the lamp and the photosynthesis fixed to control light intensity. Add sodium hydrogen carbonate to the water to ensure a constant supply of carbon dioxide. Otherwise, this could limit the rate. Leave the Elodea for 5 minutes to acclimatize at each depth before collecting the volume of gas. Then measure the rate of photosynthesis by collecting the volume of gas produced in 20 minutes using a syringe. Your time could be different. It could be 30 minutes, 40 minutes, 1 hour. It's up to you. Then repeat the process 3 times and calculate the mean values. This is 3 times maybe at each depth. Control other variables like pH using a buffer, wavelength using a color filter, temperature using a water bath, and type and age of plant using the same source of Elodea and water throughout. So this was my kind of setup. You didn't have to draw, just to show you how you could set up the experiment. And for those of you who are not good at explaining, then you gotta learn how to draw, because sometimes drawing could earn you some marks even if you didn't explain so well. Then they say describe how your results should be recorded, presented, and analyzed in order to draw conclusions from your investigation. So here we will record the results in a table with labels and units. Then we'll get results plotted in a graph. It could be a bar graph that if you just consider 2 different depths or 2 different light intensities. But if you varied more like I did, then we have to use a scatter graph or a line graph. And of course, the statistical test you use depends on how your data was presented. For those who use a bar graph, then you're going to use a t-test to compare the two. And for those of you who want to show a correlation by carrying out more experiments like at different depths, you have to use a correlation test like Spearman's. So I said record data in a table with labels and units, include columns for repeat experiments, calculate the mean rate of photosynthesis for each depth, be light intensity, plot a line graph of depth versus rate of photosynthesis and include units on that line graph, use a suitable statistical test like experiments to establish significance of the results. I've written something in red here. For those of you who just did 2 depths, you cannot use Spearman's, then you have to use a t-test. Then part D suggests 2 limitations of your proposed method. Because this is about photosynthesis, the surface area of the leaves is going to affect the experiment, and light intensity could vary in an unpredictable manner. So I said light intensity decreases with depth and could vary depending on the clarity of the water, making control difficult. The leaves could have different surface area affecting the rate, and some oxygen dissolves in the water and it will not be collected when we're calculating the rate. So this brings us to the end of question 4 as well as to the end of the whole paper. Thank you for being with us. Do not forget to subscribe. See you in the next video. Bye-bye.